Okay, so one step up from the sponges are the cnidarians. This phylum name comes from the Greek, which means nettle, and that's because they have these stinging cells. So a step up from the sponges is that these are radially symmetrical. Remember, that means it's symmetrical about a point, so you can cut it in half in many different planes, and you'll have two of the same type on both sides. They have a gastrovascular cavity, so that's an incomplete digestive tract where the sponges did not have that. Remember, if it's incomplete or gastrovascular, then there's only one opening, so food goes in, gets digested, waste comes out the same opening. Okay, we also refer to them as having a hydrostatic skeleton, so there is no hard skeleton, but they have a lot of fluid in between layers in the body, and that gives it shape. So it's not hard like you might think of with the skeleton, but it has fluid in between that kind of gives the organism some shape. So you can see on the left we have a sea anemone, and on the right is a jellyfish, and both of these are examples of cnidarian. So like I said, they have tentacles and stinging cells. The stinging cells are called cnidocytes. I'm not concerned that you know that term, but that's where the name for this particular phylum comes from. So I put a couple of pictures here. On the left you have the Portuguese man of war, which has lots of stinging tentacles. You can see it's caught a fish in them. And then up at the top right is a hydra, which is fresh water, okay, and they're almost microscopic. They're very small. And then we also have a picture here of the stinging cells that you would find within the tentacles. I did put a little video down here that you might want to take a look at, just looking at the symbiotic relationship between sea anemones and clownfish. So there are some organisms that are not bothered by the stinging cells, and that's a symbiotic relationship between a clownfish and a sea anemone, where the clownfish is protected by the sea anemone. And this is another picture from one of the books, and it just shows the layer of cells where you would find the stinging cells, and then when they're discharged you can see all those spines on them, and usually they have some kind of toxin in them, which is why it stings pretty bad. So when we get to this second step up, so we're a little bit higher, more complicated organism than a sponge, we have two germ layers. And remember, I told you germ stands for germinal beginning, and so during development they have basically ectoderm on the outside, that's one tissue layer, and endoderm on the inside. Okay, and that's a pretty low level organism. When you get up to humans, we have 3 germ layers. Okay, so this has to do with the number of different organ systems the organism can produce based on the amount of germ tissue that it has. And in terms of embryonic development, this is going to be a protostome. So the first opening in this early embryo is going to develop into the mouth. Okay, so this is showing just a generalized sea anemone. So we have the base that's usually attached to the substrate, and the tentacles stick up. They have another, not well developed, but they have a nerve net, meaning that they have nervous tissue that's spread all out through the body of the organism. There is no head end. They have just the same amount of nervous tissue all over the body, but it does help them to respond to any kind of stimulus when their tentacles are touched. And then when we look at these, there's 2 different body forms that we can see in cnidarians, the polyp form and the medusa form. So at the top here, the sea anemone, this is considered the polyp form. So if you think about an organism with its tentacles sticking up, That's the polyp form. The medusa form, which is the jellyfish down here at the bottom, is floating with the tentacles hanging down. Okay, so one is up, one the tentacles are hanging down. They have all of the same parts, but they present a little bit differently. So you do have a picture in both books kind of illustrating that. This here would be the polyp form, and this one here would be the medusa form. So I did just put a photograph from a coral reef, so you can see that there's some pretty big diversity. There's 5 or 6 or 7 different colors here of corals. They're different shapes, they're different sizes, and with corals, they have a calcium carbonate skeleton, and so basically they're little tiny polyps that are sticking out of holes within that calcium carbonate skeleton. So it's kind of a colonial organism because there's a whole bunch of polyps that will live together. And I don't know if you guys pay any attention to the news or what's happening in our world, but as temperatures are slowly raising, ocean waters temperatures are slowly rising, and that makes it very difficult for these corals to survive. And so we're seeing some of the big coral reefs that once had so much diversity, we're starting to lose them because they can't survive in those higher temperatures. So I put a couple of videos here for you to take a look at, one on the Portuguese man-of-war and one on a coral reef. And we've talked about symbioses on several occasions, but when I say that the coral reefs are having trouble surviving as temperatures get higher, remember that a coral is an animal that is in symbiotic relationship with an algae. So the algae within the coral animal is making food, and that helps that coral animal survive. And it's really the algae partner that has trouble with the higher temperatures. So the algae partner will leave the coral doesn't have enough food, and that causes the organism to die, and then you see these white calcium carbonate skeletons because there's no polyps, there's no coral organisms living in that coral skeleton. So just for perspective here, a couple of things when we're talking about jellyfish. Jellyfish are an example of the cnidarians. I put some choices for an extract from jellyfish that we use in biomedical research. So we have talked a little bit about agar. Maybe you remember that agar is an extract from the red algae. And I don't know if you've paid any attention to the news with this either, but green fluorescent protein is something that's extracted from jellyfish that is actually very easy to use to label cells when you are looking at them under the microscope. So it's been a very good tool in biomedical research. And then largest jellyfish, for perspective, this is a pretty low-level organism. But the largest jellyfish is called the lion's mane, and it is 3.6 meters. Remember, 1 meter is 3 feet, so this is more than 9 feet with 30-meter tentacles. That's a huge jellyfish, and they do have stingers, so that's not one you really want to run into in the ocean. Okay, so we've gone from sponges to cnidarians, and the next step up is the flatworms. So we're still talking about organisms that are protostomes, okay? But we have gone from radial symmetry to bilateral symmetry, which means now they're symmetrical about a line. So if we look at this flatworm here, this is a planarian. We can only cut it in half one way and get two sides that are the same. Okay, so that's bilateral symmetry. They have 3 germ layers. So during embryonic development, they have another germ layer, which means they're going to be able to make more different kinds of tissues. Okay, so if we look at this planarian here, you can see the pharynx coming from the mouth. And so in this case, this is the one opening. It still has the incomplete digestive tract. So food goes in, it gets digested down here in the digestive tract, and then waste comes out through that same opening. But we do see a step up in terms of the nervous system. So they don't have a nerve net where all the nerves are spread out evenly. They have cephalization, meaning that they have a head end. And so the brain is not like what you would think of a human brain, but it's a good number of nerve cells that are all at the same point there. And then they also have these eye spots. And again, eye spots here are not an eye that they can really focus on things, but it helps them move. And in this case, usually these are animals. They're moving away from light rather than moving towards light. So this is a pretty good little video just to show you what they look like in nature. And I'm not going to be concerned about all the different systems here, but you can see that they do have a little bit more developed nervous system. The excretory system is working kind of like our kidneys. It's not nearly as advanced as ours. And then they have the gastrovascular cavity, the incomplete digestive system with one opening. And then you can see that they also have a rudimentary reproductive system. So these organisms are a good step up from the jellyfish or the cnidarians. So some examples, and when we're in face-to-face class, we have examples of all of these in our lab, and so I usually bring them to class and pass them around so you can see what they look like. But tapeworms are in this category, okay? The head can be microscopic, but usually the long worm itself is big enough that you can see it with your eye. It takes up residence inside intestines, and so it has all of these hooks on the head so that it can attach to the inside of the intestines, and then it basically lives off of the digested food within the host in the intestines. So there are human tapeworms, there are cat tapeworms, there's dog tapeworms, there's pig tapeworms, and this is kind of gross, but in the old days, when models were trying to stay really very thin, they would actually swallow either tapeworm eggs or a tapeworm that would take up residence in their intestines, and it would use up a lot of the nutrition that they were taking in, which would keep them very thin. I don't know that that happens anymore, but that's an example of something that at one point in time people used tapeworms for a purpose. Liver fluke, so another flatworm. Obviously they live in the liver, and your book has a life cycle. I'm not worried about life cycle, but places where you would actually be exposed to the liver fluke many times in kind of anaerobic waters. And so here is a rice paddy, and basically there's an intermediate host, so it's kind of like a vector that you would get that from. So if you know that a liver fluke is an example of a flatworm or a platyhelminthes, then I'm good. A couple pictures. This is the planarian. It's freshwater. It's not a parasite. Doesn't cause any problems. This one here is a liver fluke. It is parasitic. This is an electron micrograph of a tapeworm. And then this is a flatworm that is marine, living in the ocean. It's free living. You notice it's a lot more colorful. Almost all the time, organisms living in the ocean have a lot more colorful patterns and pigments than you see in a lot of the freshwater organisms. Okay, so I'm hoping you're keeping track of this. I'm going to give you a diagram to keep track of the steps up as we go. So the next phylum we're looking at is the mollusk or the mollusca phylum, and this word means soft-bodied. Okay, so there's a little diversity in the mollusk category or phylum, but that what they have in common is this soft body that has 3 parts. So this is a very generalized diagram of a mollusk, and there's a lot of parts on there you would not have to identify. But I'm going to start at the bottom. They all have a mantle, and this is located right underneath the shell. And this, if they have a shell, the mantle would secrete the shell, but it's soft tissue, okay? They all have a foot. It's located in different places depending on what kind of a mollusk it is, but of course that would help them to move. And then they all have this visceral mass, which is basically the internal organs. And you notice that there's quite a few. So there's a heart, there's gonads, there's a stomach. So there's a lot of different internal organs that you would find in a mollusk. And that's all part of the visceral mass. So in terms of characteristics, generally speaking, when we move to the flatworms, we brought in bilateral symmetry. Okay, when we move to the mollusks, we're looking at a circulatory system. So this would be what moves the blood around, but it is an open circulatory system, meaning that the blood is not all contained within vessels. There is blood sloshing around on the inside of these organisms. We have a closed circulatory system. We do not have blood sloshing around inside of us. Okay, it's all enclosed in your heart or your vessels. The step up here is that they also have complete digestion, which means now they're going to have a mouth, and food gets digested and waste is excreted from the anus. And so there's 2 openings, which is a step up from the flatworms. So we're going to look at 4 classes based on either where you find the foot on these organisms or if they have shells. Okay, so chitons are ones that you're probably not quite so familiar with. We're going to look at some pictures, but chitons always have 8 shells that are secreted from the mantle. Okay, we'll look at pictures here in a minute. I'm just going to look at the classes. Gastropods, so gastro refers to the stomach, and podiapod refers to the foot. And so this means that these organisms have their foot located right next to their stomach. Okay, the cephalopods, remember cephalo means head, these are the ones that have the foot located right next to the head. So the foot is located in different places. And then the bivalves, bi means 2, and valve means shell. So these would all be mollusks that have 2 shells, and usually that's symmetrical in that way, where there's both 2 shells that close up and match. Okay?